Welcome back to the deep dive. We are continuing our journey through the machinery of life, and today, the investigation takes a really sharp turn. If you've been following our Medlock Holmes narrative, our sort of metaphorical detective at the crime scene of biology, you know where we've been.
We've been looking at the body, the physical evidence.
Exactly. We've examined the structure of biomolecules. We've analyzed the boundaries, those lipid membranes, and we've studied the catalysts, the enzymes that make everything happen. We've, in a way, built the engine and the chassis of the car.
Yeah.
But today, Medlock Holmes has to stop looking at the car.
And start looking at the driver.
Or at least the instructions being given
yeah.
to the driver, right?
Right. We are moving from the physical evidence to the wiretap phase.
Wiretap phase, I love that. Because today we are tackling the central mystery that, quite frankly, just blows my mind every time I stop to think about it. You are made of trillions of cells. Trillions. Most of them are locked away in the dark, deep inside your tissues, miles away from the outside world.
They can't see, they can't hear.
Not a thing.
Yeah.
And yet, if you see a tiger, your heart races, your pupils dilate, your muscles prime for action, and your digestion just shuts down, all of it, instantly.
It's the Tower of Babel problem, but solved. It's astonishing. It is.
How do trillions of blind, mute cells coordinate that kind of complex body-wide action without ever touching the source of the instruction?
Well, that is the fundamental definition of multicellular life, isn't it? Life is not just about structure, it is about information flow. Without communication, you don't have an organism, you just have a bag of confused cells bumping into each other.
So today, we are diving into biochemical signaling.
You're going to crack the code.
We are going to try. And to guide us through this labyrinth of whispers and shouts, we have our two trusty guidebooks right here on the desk.
The heavy hitters. We've got Leninger's Principles of Biochemistry, which for today is really our rule book for the grammar of this cellular language. It explains the physics, the thermodynamics, the deep logic of how signals work.
And in the other corner, Harper's Illustrated Biochemistry,
which is our field manual. Harper's takes those principles and shows us how they apply in the messy reality of the human body, the endocrine systems, pharmacology, and crucially, what happens when the signals get crossed and lead to disease.
Okay, so a quick disclaimer before we start tapping the phone lines. This deep dive is a synthesis. We're building a conceptual map here. We aren't replacing the texts. The goal is to illuminate the patterns so that when you, the listener, go back and read the chapter on biochemical signaling in Leninger or Harper's.
You're not just memorizing Greek letters and pathway names.
Exactly. We want you to see the story so the reading becomes a purposeful investigation.
Right. You'll see the forest, not just the trees.
Okay, to set the stakes, I want to start with a thought experiment. I call it the Silent City.
I like it. Set the scene for me.
Imagine a bustling metropolis with millions of inhabitants. But there's a catch. No one can speak. No one can see anyone else. They're all blindfolded and mute, isolated in their own little windowless cubicles.
That's a terrifying image.
Oh, it gets worse. Imagine a flood is coming. A massive tidal wave is approaching the city walls. Within seconds, these millions of isolated, blind, mute individuals need to coordinate a massive defense.
So they need to close the floodgates.
Reinforce the walls, evacuate the low ground. If they don't act in perfect unison, everyone dies. How on earth do they do that?
That isn't just a thought experiment. That is the challenge the human body faces every moment of every day. A spike in blood sugar, that's the flood. A drop in temperature, that's the freeze.
And the solution is biochemical signaling.
It's not magic, it's chemistry. But it's chemistry with a vocabulary, a language.
So let's break down that vocabulary. We're starting with part one, the grammar of cellular language. For this, we're drawing heavily from Leninger. Leninger outlines 6 key features of what it calls signal transducing systems. These are the rules of engagement.
These are the universal principles. I mean, whether you are a human, a fruit fly, or a fern, if you are a multicellular organism, you are using these rules.
Okay, rule number 1, specificity.
This is the classic lock and key concept, but we need to treat it with a bit more nuance than, say, high school biology. You have to realize that a cell is just swimming in a chaotic soup. It is constantly bombarded by thousands of different molecules, nutrients, waste products, signals from its neighbors, drugs.
It's like trying to listen to one specific radio station in a room where 1,000 people are screaming different things at the top of their lungs.
That's a perfect analogy. The cell has to tune in only to the signal it needs. And this is achieved through the receptor. The receptor is a protein, usually spanning the membrane, that is shaped perfectly to accept only one specific signaling molecule, which we call the ligand.
And Lenergic is a great example of this to show just how specific we're talking. Thyrotropin-releasing hormone.
Right. This hormone travels all through the blood to the pituitary gland and triggers a response there, but on its way, it bumps into millions and millions of hepatocytes' liver cells.
And the liver cells.
They completely ignore it. They don't even know it's there.
It's effectively invisible to the liver cell.
Correct, because the liver cell lacks the receptor for it. It's simply not tuned to that frequency. And this specificity it's physically measurable. This is where Leninger brings up scattering analysis and the dissociation constant, the tocodillar.
Okay, let's unpack that, because I see Kikudoleria, my eyes glaze over a little bit. What does a tecodolar of, say, 10,000 molar actually mean to me in practical terms?
Think of it as a measure of stickiness or affinity. A lower tecodolar means the receptor is incredibly good at catching very, very rare molecules. A postodolar of $10 means that even if the hormone is present in tiny amounts, we're talking nanomolar concentrations, the receptor will find it, grab it, and hold on tight.
So lower the number, the tighter the grip.
Exactly. It implies a very high affinity. The lock is exquisitely designed for that one key.
So rule 1 is specificity. Ignore the noise, find the signal. Rule number 2 involves turning that signal into action. This is sensitivity and amplification.
This is how a whisper becomes a shout, because usually the hormone itself is just a single molecule or a handful of them. One molecule binding to the outside of a cell is not, by itself, enough to change the destiny of that cell.
So how does it do it? It needs to get louder.
Through a cascade. An enzyme cascade, Leninger explains this beautifully. Imagine one hormone molecule binds to one receptor. That single activated receptor can then activate not just one, but maybe 10 or 100 G proteins inside the cell.
Okay, so 1 becomes 100.
And each of those G proteins activates an enzyme, let's say adenylyl cyclase. Each of those enzymes then creates hundreds or thousands of product molecules, like CAM-MP.
And each of those might activate another layer of enzymes.
Precisely. It's a pyramid scheme.
It is. It's a biological pyramid scheme, but, a productive one.
A very productive one. Within milliseconds, that single binding event on the surface has resulted in thousands, maybe millions of active molecules inside the cell. That is amplification.
And Leninger points out that this happens by orders of magnitude. It explains why we don't need gallons of adrenaline coursing through our veins. A tiny drop will do.
Exactly. The system is exquisitely sensitive because it can amplify so powerfully.
Okay, that brings us to rule number 3. Modularity. This one always reminds me of Legos.
That's the perfect analogy. Evolution is, it's lazy. Or rather, it's efficient. It doesn't like to reinvent the wheel for every single problem.
Right.
So signaling proteins are built from modular parts. You have these mix and match protein domains that can be shuffled around.
You're talking about things like the SH2 domain?
Yes. The SH2 domain is a classic example. It's like the sticky side of a Velcro strip that is designed to stick only to a phosphorylated tyrosine residue. If a protein has an SH2 domain, it can click into any signaling pathway that presents that specific phosphorylated hook.
So this allows cells to build incredibly diverse signaling circuits from a limited set of parts.
Mm-hmm.
You don't need a brand new protein for every single function. You just snap on a different domain.
You got it. It allows for massive complexity without needing a correspondingly massive genome. It's combinatorial.
Rule number 4 is one I think we can all relate to. Desensitization or adaptation.
This is the nose blindness of the cell.
Right. I was just thinking that, like when you walk into a room that smells of freshly baked cookies and it's amazing, but 5 minutes later you can't smell it anymore. Your nose has adapted.
The cell does the exact same thing. If a signal persists, the system has to turn down the volume. If the fire alarm is ringing constantly, eventually you have to stop treating it as an emergency.
So what does the cell do?
A couple of things. The feedback loop might kick in and chemically modify the receptor to stop it from sending the signal. Or in a more extreme case, it might physically pull the receptor inside the cell, a process called endocytosis, removing it from the surface entirely.
Leninger uses the visual of walking from bright sunlight into a dark room. Your visual system, the rhodopsin in your eyes, has to modulate its sensitivity or you'd be blinded.
And that visual system, rhodopsin, is actually a G protein coupled receptor system. It's using the very same machinery we're discussing. It's all the same principle.
That's wild.
Right.
So our senses are just a specialized form of this. Okay, rule number 5. Integration.
This is the democratic process inside the cell. A cell rarely receives just one signal at a time. It might be getting signal A from an insulin molecule saying grow and store energy, and at the same time getting signal B from an adrenaline molecule saying stop, burn energy now.
So who wins? It's a contradiction.
They have to vote. The cell mathematically sums these inputs. This is called crosstalk. It takes the positive input from receptor A and the negative input from receptor B, and the net result, whether that's a change in membrane potential or the concentration of a second messenger, determines what the cell actually does.
So the cell is a tiny computer. It's summing inputs to produce a single unified output.
Correct. It is biochemical computation happening constantly.
And finally, the sixth rule, localized response.
This is about containment. Sometimes you don't want the message to broadcast everywhere inside the cell. You want to change the chemistry at this specific synapse or on that specific patch of the membrane.
How do you keep the signal from spilling over?
Cells use scaffolds. These are large proteins that act like a workbench, holding all the necessary signaling machinery together in one clump. This keeps the message contained and ensures the response is fast and efficient, right where it's needed.
So just to check in on the big picture here, let's say this grammar fails. What if a pathway fails to amplify?
The organism's reaction time would drop to practically zero. You wouldn't be able to run from that tiger because your muscles wouldn't get the message fast enough or loud enough to act.
And what if it fails to desensitize, if the alarm never turns off?
You get chaos. Uncontrolled signaling. That's essentially what happens in many drug addictions, or in more biological terms, something like a seizure or, as we'll see later, cancer. The off switch is as important as the on switch.
All right. That's the grammar, the fundamental rules. Now let's talk about the messages themselves. We're moving to part 2, the diversity of messengers, and for this, we're picking up Harper's Illustrated Biochemistry.
Yeah, Harper's gives us a really clean way to think about this. It categorizes hormones into 2 main tribes. And this distinction is crucial because it tells you how the hormone works just by knowing what it's made of.
We've group 1 and group 2. Let's start with group 1, the lipophilic intruders.
These are your steroids like testosterone and cortisol, your thyroid hormones, your retinoids. The key characteristic here is that they are lipophilic, fat-loving, or hydrophobic.
Which means, in terms of getting into the cell,
it means that the cell membrane, which is a lipid bilayer, is not a barrier to them. They don't need to knock on the door. They just walk right through the wall.
so they act like ghosts. They can just float through the plasma membrane and go straight into the cell cytoplasm.
Exactly. Their destination isn't the cell surface. It's usually a receptor protein floating inside the cytoplasm or even waiting for them directly inside the nucleus. Once the hormone binds to this intracellular receptor, the whole complex acts directly on the DNA.
No.
They bind to specific DNA sequences called hormone response elements, or HREs.
So they are literally changing the blueprint.
They are acting as architects. They enter the building, unroll the blueprints, the DNA, and say, okay, we need to build a new wing. They directly change gene expression. They trigger the transcription of new mRNA, which leads to the synthesis of new proteins.
And because they're building entirely new infrastructure, this is going to take some time, right?
Yes. This is a slow, sustained change. It can take hours or even days to see the full effect of a steroid hormone, but that effect lasts for a long time.
Harper's mentions a really cool clinical detail here about how glucocorticoids get into the nucleus. It involves something called heat shock proteins.
Right, the heat shock proteins, like HSP90. It's a great example of regulation. When the glucocorticoid receptor is just sitting in the cytoplasm waiting, it's bound to a chaperone protein, HSP90. This chaperone physically covers up the nuclear localization signal on the receptor.
So it's like a bouncer blocking the door to the VIP room.
That's a good way to put it. It keeps the receptor from entering the nucleus prematurely. But when the hormone binds to the receptor, it causes a shape change that makes the HSP90 fall off. The ticket is revealed, and the receptor can now march right into the nucleus and get to work.
So the VIP pass was in its pocket the whole time. It just needed the hormone to show it.
Exactly.
Okay, that's group 1, the architects. Now what about group 2, the hydrophilic couriers?
These are your peptides, like insulin, proteins like growth hormone, and catecholamines, like epinephrine or adrenaline. They are all water-soluble.
Which means they bounce right off the oily lipid membrane.
Correct. Oil and water don't mix. They cannot enter the castle. They have to knock on the front gate. They bind to cell surface receptors.
So if they can't get in, how do they actually deliver the message?
They rely on second messengers. The hormone itself is the first messenger. It's the one that knocks on the door. The receptor then activates a second messenger inside the cell molecules, like cyclic AMP or calcium, to run the message up to the throne room and tell the cell what to do.
So if group I hormones are architects changing the blueprints.
Group 2 are dispatchers. They are standing outside the factory window, shouting orders to the workers on the floor. Turn on this machine, turn off that conveyor belt, speed up production.
And because they're just toggling existing machines, just changing the activity of proteins that are already there.
It is fast, incredibly fast. We're talking seconds to milliseconds. This is your fight or flight response. You don't have time to build new proteins when a bear is chasing you. You need to activate the enzymes you already have right now.
That distinction, architects versus dispatchers, is so incredibly helpful. It just clicks.
And it explains so much about pharmacology, too. Group I drugs, like steroids, usually take a while to work. Group 2 drugs, or drugs that mimic them, can be almost instantaneous.
Let's dig deeper into the machinery of Group 2 then. This brings us to Part 3, the GPCR, the G-protein coupled receptor. This seems to be the universal switch of biology.
It really is. It's the largest single family of cell surface receptors in humans. There are over 800 of them, and the structure is absolutely iconic. We often call it the heptahelical receptor.
Hepta meaning 7.
Yes. Picture a protein that looks like a snake threading its way back and forth through the cell membrane 7 times. That's why it's also called a serpentine receptor.
And hooked onto this snake on the inside of the cell is the star of the show, the G-protein.
Exactly. The G protein is a trimeric complex that has 3 parts, an alpha subunit, a beta, and a gamma. And this is where the switch metaphor becomes quite literal.
Okay, walk us through the switching mechanism. How does it turn on and off?
Okay. In the inactive state, when the switch is OFF, the alpha subunit is holding on to a molecule of GDP, guanosine diphosphate.
GDP, got it.
When a hormone, the ligand, lands on the receptor on the outside of the cell, it's like a hand fitting into a glove. It causes the receptor to change its shape. The shape change sort of nudges the G protein on the inside, causing the alpha subunit to let go of its GDP and in its place pick up a GTP guanosine triphosphate.
So the GDP is exchanged for GDP.
And that simple swap is the on-end signal. The alpha subunit, now holding GTP, gets a new shape and it detaches from the beta-gamma pair. It's now free to go off and do its work. It slides along the inside of the membrane to find its target enzyme.
But switches need to be able to turn off too. That was Rule 4, desensitization.
A crucial point. The alpha subunit has a built-in timer. It has intrinsic GTPase activity, which is a fancy way of saying it can slowly chop that third phosphate off of GTP, turning it back into GDP.
So it resets itself.
It resets itself. Once it does that, it turns itself off, refines its beta-gamma partners, and the whole complex is ready for the next signal. It's a beautifully elegant, self-limiting switch.
Let's apply this to a real-world example. The book talks a lot about the CONMP pathway, the classic beta-adrenergic system. This is the adrenaline rush, right?
This is the textbook case.
Okay.
So epinephrine binds to the beta-adrenergic receptor on, say, a muscle cell. The switch flips. The G's protein, the S is for stimulatory, grabs GTP. The alpha subunit then breaks away and slides over to an enzyme embedded in the membrane called adenyl cyclase.
Adenyl cyclase.
This is the amplifier. Adenyl cyclase takes ATP, the cell's energy currency, and starts cranking out a molecule called cyclic AMP, or KMP, tons of it.
So KMP is the second messenger.
That's our second messenger. It's the dispatcher's message being run inside the factory. AMP then floats through the cytoplasm and finds its target, protein kinase A, or PKA.
Now, Leninger has a fascinating detail about how PKA works. It's almost like it involves hostages.
In a way, yeah, that's a good way to think about it. PKA is made of 4 parts. 2 catalytic subunits, these are workers that do the phosphorylating, and 2 regulatory subunits, these are the guards. In the inactive state, the guards are holding the workers hostage, preventing them from doing anything.
And KMP is the negotiator, or the ransom.
It's the ransom. CMP molecules bind to the guards, the regulatory subunits. This binding causes the guards to change shape and let go of the workers. The catalytic subunits are now free to roam the cell and phosphorylate their targets.
And phosphorylating things means adding phosphate groups to them, which acts like a switch to turn them on or off.
Exactly. Mobilizing glucose from glycogen, increasing heart rate, dilating your bronchioles so you can breathe better. All of that downstream action comes from PKA being set free.
I love the thought experiment Leninger mentions here about Fenody. It sounds like a way to actually watch this happening in real time.
FRE is amazing. It stands for fluorescence resonance energy transfer. Scientists can be very clever. They can tag the regulatory subunit, the guard, with a blue fluorescent protein and the catalytic subunit, the worker, with a green one.
So when they're hugging each other, when the hostage situation is ongoing, they're very close together.
Right, so close that if you shine a blue light on the cell, the blue protein absorbs it, and instead of emitting blue light back, it transfers that energy to the nearby green protein, which then emits green light. You shine blue, but you see green.
But what happens when adrenaline hits?
The CAMP floods the cell, binds to the guards, and pulls them away from the workers. They separate. Now, when you shine blue light on the cell, the energy transfer can't happen. The blue protein just emits blue light. You can literally watch the color of the living cell shift from green to blue as the adrenaline signal propagates.
That is just incredibly cool. We can actually see the chemistry thinking.
And you can. It's beautiful.
But, as with all things, this beautiful system can break. And Harper's gives us a terrifying example of a broken switch. Cholera.
Cholera is a brutal but very effective teacher of biochemistry. The cholera toxin is an enzyme that gets into our intestinal cells and attacks the G's protein.
The very same G's protein that stimulates cell AMP.
The very same one. The toxin chemically modifies the alpha subunit so that it loses its timer. It completely breaks the GTPase activity. It cannot hydrolyze GTP back to GDP.
So the switch is stuck, is jammed in the on position.
Permanently on in. Adenyl cyclase never stops. It just churns and churns. CMP levels in the cell skyrocket to insane levels. pKa goes into overdrive.
And what does all that hyperactive pKa do in the gut?
Well, one of pKa's many targets in the intestinal lining is a chloride channel, specifically the CFTR channel, the same one implicated in cystic fibrosis. pKa phosphorylates it and forces it to stay open. Chloride ions pour out of the cell into the gut.
And where salt goes?
Water follows. Osmosis. Massive amounts of water are pulled from the blood and tissues into the intestine to follow the salt. You get catastrophic, life-threatening diarrhea. The patient dies not from the bacteria itself, but from simple, profound dehydration.
So it's a signaling failure, a communication breakdown.
More specifically, it's a failure of termination. It proves in the most dramatic way possible that the ability to turn a signal off is just as vital as the ability to turn it on.
Wow. Okay, so that's the TMP pathway, a huge player. But Harper's reminds us there's another major pathway for these group 2 hormones. This brings us to part 4. Second messengers beyond CAMP, the phosphonocetide system.
A bit of a mouthful, but absolutely crucial. This is the other major GPCR path.
So instead of a GS protein, we have a GQ protein.
Yes, GQ. And instead of activating adenyl cyclase to make CAMP, GQ activates an enzyme called phospholipase C, or PLC.
I like the name lipase. It implies it's cutting lipids.
That's exactly what it does. Its target is a specific phospholipid in the membrane called PIP2. PLC chops it into 2 pieces, and this is the brilliant part. It creates 2 second messengers at once. One piece stays in the membrane, and one piece floats away into the cytoplasm.
So you get 2 messengers for the price of 1 signal.
Precisely. The piece that floats away into the cytoplasm is called IP3, or inositol trisphosphate. You can think of it as the key. It floats over to the endoplasmic reticulum, which is a huge storage vault for calcium, and it unlocks calcium channels there.
So calcium floods out into the cell.
A massive spike in cytosolic calcium, which is a powerful signal in itself. It activates all sorts of things. Meanwhile, the other piece that was left behind, DGA, or diacylglycerol, acts as the anchor. It stays in the membrane and serves as a docking site to recruit another enzyme, protein kinase C, or PKC.
So with one signal, you get a calcium spike and PKC activation.
Right, and this system controls a whole different set of things like smooth muscle contraction, inflammation, and neurotransmitter release.
And Harper's makes a fascinating clinical connection here to bipolar disorder.
Yes, with lithium.
Yeah.
For decades, we've known lithium is an effective mood stabilizer, but the mechanism was a bit of a mystery. It turns out one of its main actions is to interfere with the recycling of inositol. It essentially prevents the cell from regenerating the PIP2 that PLC needs to cut.
So it dampens this specific signaling pathway.
It quiets the noise. It turns down the volume on the IP3DD pathway, which seems to be overactive in the brains of people with bipolar disorder.
So again, pharmacology is just getting in there and tweaking the signal strength.
It always is.
Okay, moving on to part 5. We've talked about these GPCRs, which are like buttons that call for help from a G protein. Now let's talk about a different class, the builders. the receptor tyrosine kinases, or RTKs.
This is a fundamentally different mechanism. GPCRs are just switches. RTKs are more than that. They are enzymes themselves.
So the receptor is the enzyme. It's not just calling for one.
Correct. The part of the receptor that sticks out inside the cell has intrinsic kinase activity. Specifically, it's a tyrosine kinase. It adds phosphates to tyrosine residues.
And the classic example here is the insulin receptor.
Insulin is the poster child for RTKs. When an insulin molecule binds to the outside, it doesn't just flip a switch. It causes two receptor molecules to come together to pair up or dimerize. And then the two receptors phosphorylate each other.
That's autophosphorylation. It's like they high-five to kick themselves into gear.
It's exactly like that. This cross-phosphorylation creates a fully active enzyme complex, and the phosphorylated tyrosines act as a scaffold. They become a docking station for other proteins. In the case of insulin, it recruits a big adapter protein called IRS1, the insulin receptor substrate.
And this IRS1 is like a power strip. You can plug other things into it.
A very sophisticated power strip. It gets phosphorylated itself, creating even more docking sites. This allows proteins with those SH2 domains we mentioned earlier, like a protein called GRB2, to plug in. And this builds a bridge to a famous cascade.
The MAPZ cascade.
The Ras-Raf-Mek-ERK pathway, a signaling superhighway that goes all the way to the nucleus to change gene expression, telling the cell to grow, to divide, and of course to take up glucose from the blood.
Now here is where it gets really, really interesting to me. The concept of crosstalk that Leninger brings up. The battle between insulin and epinephrine.
It's a fundamental physiological debate, right? Epinephrine is screaming, burn energy, we have to fight the bear. And insulin is calmly saying, store energy, we just ate a donut. They can't both be right at the same time.
So how does the body settle that argument?
Through biochemical prioritization. The signaling pathway from the insulin receptor can actually reach over and directly phosphorylate the beta-adrenergic receptor, the adrenaline receptor.
Wait. So the insulin system physically attacks the adrenaline receptor.
It tags it. It puts a phosphate on it. And that phosphate tag is a signal for the cell to internalize the adrenaline receptor, to pull it off the surface and hide it inside.
So insulin doesn't just try to shout louder than adrenaline. It literally hides adrenaline's microphone so it can't be heard.
Exactly. It removes the competition. It's a beautiful example of brutal efficiency. The body decides that in that moment, storage is the priority, so it actively deafens itself to the mobilization signal from adrenaline.
That integration is incredible. But it also brings us to a darker side of signaling. Part 6. When signaling fails. We've touched on cholera, but Harpers and Leninger both make it clear that cancer is fundamentally a disease of signaling.
It absolutely is. Cancer is what happens when the cellular conversation goes profoundly wrong. And the prime suspect in so many of these cases is a protein called RAS.
RAS. This is that small G protein in the insulin and growth factor pathway we just talked about.
Right. RAS is the most commonly mutated oncogene in all human cancers. Remember how we said the G protein has a built-in timer? It hydrolyzes GTP back to GDP to turn itself off.
The self-limiting switch.
In about 30% of human cancers, the RAS protein has a mutation that breaks that timer. It can grab GTP and turn on in, but it can never, ever turn itself off.
So it's like the cholera toxin. It's a switch that's stuck on it. It's just screaming, divide, grow, divide, come to a full threat.
Continuously. Even if there is no growth factor outside at all, the cell becomes deaf to the silence from the outside world because of the constant noise on the inside.
But surely there are safeguards for this, right? The brakes of the system.
There are. We call them tumor suppressors. Harpers discusses two of the most famous ones, P53 and the retinoblastoma protein, PRB.
I found the retinoblastoma story both fascinating and really tragic.
It's a classic example of what we call the two-hit hypothesis. RB is a brake pedal. Its job is to stop the cell cycle at a critical checkpoint. Now, if a child inherits one broken copy of the ARB gene from a parent, they are primed. They're born with only one working brake pedal in all their cells.
So if a random mutation, just bad luck, hits that one remaining good copy in a single retinal cell,
the brake is gone. The cell can no longer stop at the checkpoint. It just pushes through and divides and divides and divides uncontrollably. This creates the tumor in the retina that gives the disease its name.
So cancer is basically a car with a brick on the accelerator. That's the oncogene like RAS. And the brake lines have been cut. That's the tumor suppressor like RB.
That is the perfect analogy. And almost all modern cancer therapy is about trying to fix those signals-finding drugs that can take the brick off the accelerator or somehow restore the function of the brakes.
This brings us to our conclusion. My head is spinning a bit. We've traveled all the way from the cell membrane to the nucleus. We've seen the fast track of the GPCRs and their second messengers, and the slow, deliberate track of the nuclear receptors.
We've seen that life, moment to moment, hangs in the balance of these intricate chemical conversations.
And stepping back to our Medlock Holmes narrative, I think the big realization for our detective here is that biology isn't just about the presence or absence of a molecule. It's about context.
Context is everything. Think about epinephrine. It's the exact same molecule. If it hits a beta-adrenergic receptor in your heart beats faster and stronger. But if that same molecule hits an alpha-adrenergic receptor in the blood vessels of your gut, they constrict and digestion slows to a crawl.
So it's the same key, but it's unlocking two completely different doors, leading to rooms with totally different functions.
And those doors, the receptors, are coupled to different G proteins inside. A new dollar in the heart, a dollar for inhibitory in the gut. The molecule itself doesn't carry the meaning. The circuit carries the meaning. The wiring of the cell determines the reality of the signal.
Which leads me to this final provocative thought for our listeners. We've spent this whole time talking about how cells listen. But I want you to think about how you are listening to this deep dive right now.
Go on.
Well, your ears are translating physical vibrations into nerve impulses. But what about your other senses? Your eyes. The rhodopsin that detects light is a GPCR, your nose. The olfactory receptors that detect smells are all GPCRs, your tongue. Many of your taste receptors for sweet, umami, and bitter, also GPCRs.
I see exactly where you're going with this.
The machinery that we as conscious beings use to perceive reality, to see a sunset, to smell a rose, to taste a coffee, it's the exact same molecular machinery that a liver cell uses to detect adrenaline or insulin. Our entire perception of the outside universe is just biochemical signaling.
So it raises the question, is reality, as we experience it, just a very specific, highly organized concentration of CAMP in your brain?
A question to ponder. Maybe for a sleepless night.
Indeed.
That's it for this deep dive into biochemical signaling. We've cracked the wiretap. Next time, we move from the communication network into the engine room energy and regulation.
From the software to the power grid, I can't wait.
See you then.